Hello and welcome to another installment of General Biology. What we're going to get into is Chapter 6 of Campbell Biology, A Tour of the Cell. The fundamental units of life. All organisms are made of cells. The cell is the simplest collection of matter that can be alive. All cells are related to their descent from earlier cells. Cells can differ substantially from one another, but share many common features. Biologists use microscopes and the tools of biochemistry to study cells. Cells are usually too small to be seen by the naked eye. Microscopes are used to visualize cells. In a light microscope, visible light is passed through a specimen and then through glass lenses. The lenses refract or bend the light so that the image is magnified substantially. Three important parameters of microscopy: magnification, the ratio of an object's image size compared to its actual size; resolution, the measure of the clarity of the image or the minimum distance of two distinguishable points; contrast, which is visible differences in brightness between parts of a sample. What this is meant to show is that the eye can see things from our height all the way down to a frog egg. A light microscope is good for getting down to plant and animal cells and their organelles, as well as bacteria. Super resolution microscopy can even get down to smaller bacteria and ribosomes, and electron microscopes are able to get all the way down to proteins and lipids, and in some cases powerful enough to even see atoms with a powerful enough electron microscope. Power of electron microscopy is, if it's bigger than an electron, it's able to be eventually visible by an electron microscope. Light microscopes can magnify effectively to about 1,000 times the size of the actual specimen. Various techniques enhance contrast and enable cell components to be stained or labeled, essentially adding a different fluid to your specimen to help certain parts of a cell or certain components become distinguished, whether the nucleus by adding a contrast that attaches to DNA, or certain organelles by adding a contrast that attaches to proteins. The resolution of standard light microscopy is too low to study organelles, though. Therefore, membrane and closed structures in eukaryotic cells need something more powerful. What we're seeing here is different resolutions and different techniques used to get differing images of specimens. Those such as light microscopy, from the bright field technique, which can have unstained or stained specimens. Staining, again, usually helps contrast or add distinguished parameters. And then there's phase contrast and differential interference contrast by Minorsky. Usually these units we're looking at micrometers or micrometers. A micrometer is 10 to the negative 6 meters. Depending on the contrast or the technique, You can get various different beautiful images, and sometimes one initial scan can go through deconvolution to get a more distinguished image that's using computational techniques. Scanning electron microscopy, or SEM, transmission electron microscopy, TEM, those are able to get down to, those can distinguish further than typical light microscopy. But more powerful light microscopy includes super resolution. Here is how far can we look into a small sample and how well can we distinguish the different traits. Two basic types of electron microscopes, EMs, are used to study subcellular structures. There's the scanning electron microscope, which has a focus on beam, which focuses a beam of electrons onto the surface of a specimen, providing images that look three-dimensional. Then there's transmission electron microscopy, which focuses a beam of electrons through a specimen. TEMs are used mainly to study the internal structure of cells. Recent advances in light microscopy have resulted in labeling individual cells with fluorescent markers to improve the level of detail that can be seen. Confocal microscopy and deconvolution microscopy provide sharper images and 3D tissues and cells. And then new techniques for labeling cells improve resolution, and super resolution microscopy allows one to distinguish structures as small as 10 to 20 nanometers, a nanometer being 10 to the minus 9th meters. Cell fractionation takes cells apart and separates the major organelles from one another. Centrifuges fractionate cells into their components by spinning a sample at high speeds. Cell fractionation enables scientists to determine the functions of organelles. And in biochemistry and cytology, the study of the contents of cytoplasm help correlate cell function with structure. Here we're looking at example of centrifugation, separating a blended sample or a mixed sample through homogenization, and then centrifuging them to help separate different densities of sample. The G being how many times the standard force of gravity is being plugged in. And sometimes higher G's for longer periods of time can result in better separation, depending on what is preferred. The pellet at the bottom is usually what is either desired or removed, depending on what is trapped in it. And then essentially the liquid portion, that's where we're removing stuff from to form the pellet at the bottom, which may consist of different components. The basic structural and functional unit of every organism is one of two types of cells, prokaryotic or eukaryotic. All organisms of the domains of bacteria and archaea consist of prokaryotic cells. Protists, fungi, animal cells, and plant cells all consist of eukaryotic cells. Basic features of a cell is the plasma membrane that surrounds the cell, the semi-fluid substance called cytosol inside the cell, chromosomes, which are how genes are packaged, and ribosomes, which are used to make proteins. Prokaryotic cells are characterized by having no nucleus to store their DNA. Their DNA usually exists as an unbound region called the nucleoid. There is no membrane-bound organelles inside a prokaryotic cell, and cytoplasm is bound by the plasma membrane. Different types of bacterial cells can include rod-shaped bacterium. Eukaryotic cells, they will have DNA protected by a membrane-bound nucleus, specifically a double membrane, adding extra protection from outside changes that could mutate or harm that DNA structure. Eukaryotic cells also have membrane-bound organelles, and they have cytoplasm in the region between the plasma membrane and nucleus. Eukaryotic cells are generally much larger than prokaryotic cells. The plasma membrane is a selective barrier that allows sufficient passage of oxygen and nutrients and waste to service the volume of every cell. Through transmission electron microscopy, looking at the outside of a cell, transferring into the membrane, we see hydrophilic regions, and in the center a hydrophobic region, and transport proteins latched on inside, and sometimes proteins can have carbohydrate side chains on top of them. Also phospholipids, which are which makes up the membrane. Metabolic requirements set upper limits on the size of cells. The surface area to volume ratio of a cell is critical. As a cell increases in size, its volume grows proportionally more than its surface area. Effectively, the distance between the insides of a cell from one organelle to another increases much more fast than the outside of a cell, which is what is used to transport stuff in and out. A eukaryotic cell has internal membranes that divide the cell into compartments called organelles, essentially organs of a cell. The basic fabric of biological membranes is the double layer of phospholipids and other lipids. Plant and animal cells have most of the same organelles. There's the endoplasmic reticulum, which includes the rough ER and the smooth ER. Rough ER having more ribosomes attached to them. There is also the Golgi apparatus, lysosomes, mitochondrion, the plural or the singular of mitochondria, the powerhouse of the cell. Can't have a biology class without saying that at least once. And many other factors as well. The nucleus, which includes the nuclear envelope around it, the nucleolus, which is the center, and then the inter-between area of chromatin, genetic material. Cytoskeleton, which has structure through microfilaments, intermediate filaments, and microtubules. Microvilli, which are on the outside of a cell. Peroxisomes, which can help digest stuff because they contain peroxides in them. Many, many things. A eukaryotic cell has, went forward a bit. Here we have just a plant cell now, where the prior example was an animal cell. What we see here as difference is the addition of a central vacuole. Plasmodesmata, the central vacuole, which essentially has a lot of fluids. The cell wall, which adds firm structure. In addition to the cell membrane. And then many of the same things as well. The nucleus contains most of the DNA in a eukaryotic cell, protecting it from outside stimuli. Ribosomes use information from DNA to make proteins. The nucleus contains most of the cell's genes and is usually the most conspicuous organelle. The nuclear envelope encloses the nucleus, separating it from the cytoplasm. The nuclear envelope is a double membrane. Each membrane consists of a lipid bilayer. What we see is a pore complex, which is where mRNA strands can leave the nucleus. Pores line with a structure called a pore complex regulate the entry and exit of molecules from the nucleus. The nuclear size of the envelope is lined by the nuclear lamina, which is composed of proteins and maintains the shape of the nucleus. In the nucleus, DNA is organized into discrete units called chromosomes. Each chromosome contains one DNA molecule associated with proteins called chromatin. Chromatin condenses to form discrete chromosomes as a cell prepares to divide. The nucleolus is located within the nucleus and is the site of ribosomal RNA synthesis, or the production of rRNA. Ribosomes are complexes made of ribosomal RNA and protein. Ribosomes carry out protein synthesis in two locations: in the cytosol, where they are called free ribosomes, and on the outside of the endoplasmic reticulum, or the nuclear envelope, where they are called bound ribosomes, bound to the membrane surface of those two organelles. Ribosomes consist of a large subunit and a small subunit, both of which come together to help produce proteins. The endomembrane system consists of the nuclear envelope, endoplasmic reticulum, or ER, the Golgi apparatus, the lysosomes, vacuoles, and plasma membrane. These components are either continuous or connected via transfer of vesicles. Vesicles are essentially different couriers that shuttle material from one organelle to another. The endoplasmic reticulum accounts for more than half of the total membrane in many eukaryotic cells. The ER membrane is continuous with the nuclear envelope. They are attached together. There are two distinct regions of the ER: the smooth ER, which lacks ribosomes, and the rough ER, whose surface is studded with ribosomes. Essentially, portions of it can come off as vesicles or transport vesicles, which are used to carry material between different organelles. The smooth ER has the following functions. It will synthesize fats or lipids. It will metabolize sugars, carbohydrates. It helps detoxify drugs and proteins, as well as stores calcium ions. I wasn't aware of its detoxification properties. That's pretty cool. Wow. The rough ER has bound ribosomes, which secrete glycoproteins, proteins covalently bounded to carbohydrate sugars. It distributes transport vesicles, which are and secretory proteins, which are secretory proteins surrounded by a membrane. and is a membrane factory for the cell to produce other membranes. The Golgi apparatus consists of flattened membrane sacs called cisternae. The Golgi apparatus modifies the initial products of the endoplasmic reticulum, and then it will manufacture certain macromolecules and sorts and packages material into transport vesicles. With a cis face, the Golgi apparatus has a receiving end, sort of like a packing site, has a receiving end called the cis face, and then the trans face, which is the shipping end, where it gets sent out to the rest of the cell. Essentially, the endoplasmic reticulum and Golgi apparatus are sort of like a production, manufacture, and processing working together. A lysosome is a membranous sac of hydrolytic enzymes that like to digest macromolecules. Lysosomal enzymes work best in the acidic environment inside the lysosome, essentially making a separate part of a cell that can be acidic, but keeping the rest of the cell not at those pH conditions. Hydrolytic enzymes and lysosomal membranes are made by rough ER and then transferred to the Golgi apparatus for further processing. Some types of cells can engulf another cell by phagocytosis. This forms a food vacuole. A lysozyme fuses with the food vacuole and digests the molecules on the inside. Lysosomes also use enzymes to recycle the cell's own organelles and macromolecules, a process called autophagy. Bottom left and right is essentially how, when food material gets close to the cell, a portion of its cell membrane can pinch off to form its own food vacuole, which can then fuse with the membrane of the lysosome, releasing the digestive enzymes in it, digesting the material of food, which can then be transported where necessary. Or, when it comes time to recycle a organelle that's no longer functioning, peroxisomes can enter, and as well as lysosome enzymes, helping digest the material, and then it can be reused later. Fascinating. Apparently the peroxisome was a damaged organelle that was being digested as well. Nice. Vacuoles are large vesicles derived from the endoplasmic reticulum and Golgi apparati. Vacuoles perform a variety of functions in different kinds of cells. They're food vacuoles that are formed by phagocytosis. Contractile vacuoles, found in many freshwater protists, pump excess water out of the cell. And then there are central vacuoles found in many mature plant cells that hold organic compounds and water. The endomembrane system is a complex and dynamic player in the cell's compartmental organization. Allowing for unique points in the cell to have different functions. The mitochondria, those are sites of cellular respiration, a metabolic process that uses oxygen to generate the energy molecule adenosine triphosphate. Chloroplasts found in plants and algae are the sites of photosynthesis. Peroxisomes are oxidative organelles. Now, mitochondria and chloroplasts have similarities with bacteria. They are enveloped by a double membrane. They contain free ribosomes and circular DNA molecules, separate from the nuclear DNA of cells. They grow and reproduce somewhat independently in cells. These similarities led to the endosymbiote theory. This theory suggests that an early ancestor of eukaryotes engulfed an oxygen-using non-photosynthetic prokaryotic cell. The engulfed cell formed a relationship with the host cell, becoming an endosymbiote. The endosymbionts evolved into mitochondria. At least one of these cells may have taken up a photosynthetic prokaryote, which evolved into the chloroplast organelle. Essentially, a larger cell taking up two smaller cells, or a smaller cell, but not digesting them, leading to awesome functions, where the mitochondria was able to produce more energy than it needed specifically, and chloroplast could obtain energy from the sun and process it into glucose sugar by combining carbon dioxide and water with the energy obtained from the sun. Mitochondria are found in nearly all eukaryotic cells. They have a smooth outer membrane and an inner membrane folded into cristae. The inner membrane contains two compartments, intermembrane space and the mitochondrial matrix. Some metabolic steps of cellular respiration are catalyzed in the mitochondrial matrix. Cristae represent a large surface area for enzymes that synthesize ATP. The power of mitochondria is that the folds cause substantial increase in surface area, which means that there are substantial places for ATP synthesis to occur. The mitochondrial DNA is unique in that it is separate from nuclear DNA. In the case of humans, our nuclear DNA comes half from one parent and half from the other, whereas the mitochondrial DNA is part of the egg cell and not part of the sperm cell, which is why our mitochondrial DNA comes from our mothers, whereas our nuclear DNA comes from our fathers and mothers. Chloroplasts contain the green pigment chlorophyll, as well as enzymes and other molecules that function in photosynthesis. Chloroplasts are found in leaves and other green organs of plants and in algae. What we see is that chloroplasts contain these coils called the thylakoids and intermembrane space. They have their own set of DNA, like mitochondria. There are also stroma with inner and outer membranes, and granum. Chloroplast structure includes thylakoids, which are membranous sacs stacked to form a granum. And then there are stroma, which is the internal fluid. The chloroplast in one of the group of plant organelles called plastids. The chloroplast is one of a group of plant organelles called plastids. Peroxisomes are specialized metabolic compartments bounded by a single membrane. Inside that membrane, peroxisomes produce hydrogen peroxide and convert it to water. Peroxisomes perform reactions with many different functions. How peroxisomes are related to other organelles is still being looked into. How fascinating. The cytoskeleton is a network of fibers that organizes structures and activities in the cell. The cytoskeleton is a network of fibers extending throughout the cytoplasm. It organizes the cell structure and activities, anchoring many organelles in place. It is composed of 3 types of molecular structures: microtubules, microfilaments, and intermediate filaments. Wow. A lot of publisher videos. The cytoskeleton helps to support the cell and maintain its shape. It interacts with motor proteins to produce cell motility. Inside the cell, vesicles can travel along tracks provided by the cytoskeleton. Now, a key difference between motility and mobility. Mobility with a B means that that object can be moved. Motility with a T means that that object can be moved, but it can also be the one that causes its own motion. It can move itself. A chair is mobile, but it will never move itself, whereas we as humans, we are motile. We can be moved, but we can also cause ourselves to move. These motor proteins travel along vesicles, walk vesicles along cytoskeletal fibers. How fascinating. Three main types of fibers make up the cytoskeleton. Microtubules are the thickest of the 3 components of the cytoskeleton. There are microfilaments, also called actin filaments, which are the thinnest. And then there are intermediate filaments, which are fibers with diameters in the middle range. Looking at a different structure, microtubules are hollow. Microfilaments, or actin filaments, have 2 intertwined strands of actin. Intermediate filaments are fibrous proteins coiled in cables. As far as the main functions, microtubules or tubulin polymers, they maintain the cell shape. They add compression-resisting girders essentially to the cell, allowing it to keep its shape. They help in cell motility in structures called cilia or flagella to move the cell, as well as help in the movement of chromosomes during cell division, taking genetic material to opposite sides of a cell as it reproduces into 2 new cells. The maintenance of cell shape, tension-bearing elements, changes in cell shape. Microfilaments are for maintaining cell shape. They add tension-bearing elements, which helps in changes in cell shape, muscle contraction, cytoplasm, like streaming in plants, cells, cell motility as in amoeboid movement, and division in animal cells. And then intermediate filaments are again used for cell shape. They add tension-bearing elements. They anchor the nucleus and other cell organelles, and they are part of the formation of nuclear lamina. Fascinating. Microtubules are hollow rods, 25 nanometers in diameter and about 200 nanometers to 25 microns in length. Microtubules are constructed of dimers of tubulin. Functions in microtubules include shaping the cell, guiding movement of organelles, and separating chromosomes during cell division. Usually most instructors only care about the functions, not so much the actual length and parameters. So keep that in mind. And if you want, subscribe and like the video, just because I don't think I've ever mentioned that before, and it'd be fun. Centrosomes and centrioles in animal cells, microtubules, grow out from a centrosome near the nucleus. In animal cells, the centrosome has a pair of centrioles, each with 9 triplets of microtubules arranged in a ring. Cilia and flagella. Microtubules control the beating of flagella and cilia, microtubule containing extensions that project out from the cell. Many unicellular eukaryotes are propelled through water by cilia or flagella. Both differ in their beating patterns. Motion of flagella allows for the direction of swimming used in sperm, and then the motion of cilia, direction of an organism's movement, sort of like power strokes, as opposed to a swimming tail. Cilia, like the ones found in our lungs or in our bronchi. Oh, cool. We use both. Nice. How fascinating. So many videos. No access. Cilia and flagella share a common structure, a group of microtubules sheathed by an extension of the plasma membrane, a basal body that anchors the cilium and flagellum, a motor protein called dynein, which derives the bending movements of the cilium and flagellum. Now, a lot of these proteins are usually more stressed in a course called cellular and molecular biology, genetics, or sometimes to a small extent biochemistry. So for general biology, let's just don't stress every single thing that gets mentioned down to nitty-gritty enzymes. Nice. Looking good. Slide 151 of 215. Cool. Hey, nice little star of a basal body. Oh, that's cute. Nice. Dynein has 2 feet that walk along microtubules. In my opinion, these look like penguins with wet pants trying to walk. One foot maintains contact, while the other releases and then reattaches 1 step farther along. Movement of the feet causes the microtubules to bend rather than slide, because the microtubules are held in place. Microfilaments are solid rods about 7 nanometers in diameter, built as a twisted double chain of actin subunits. A network of microfilaments helps support the cell shape. They form a cortex just inside the plasma membrane to help support the cell's shape. Bundles of microfilaments make up the core of microvilli of intestinal cells. Microfilaments that function in cellular motility contain the protein myosin. In addition to actin, cells crawl along a surface by extending pseudopodia, cellular extensions, and moving toward them. Cytoplasmic streaming is a circular flow of cytoplasm within a cell, driven by actin-myosin interactions. Plenty of information. Other different protein structures. Actin, myosin. cytoplasmic streaming in image C, amoeboid movement through the extending pseudopodium. All right. I do like that some of these slides are really just pictures that were already explained, and various videos that are only accessible through the actual book, but many different substitutes can be found online without looking too hard. Then there are the intermediate filaments, which range in diameter, not really too important, but 8 to 12 nanometers, larger than microfilaments but smaller than microtubules. Intermediate filaments are more permanent cytoskeleton fixtures than the other two classes. They support the cell shape and affix organelles in place. Most cells synthesize and secrete materials that are external to the plasma membrane. These extracellular materials and structures are involved in a great many cellular functions. The cell wall is an extracellular structure that distinguishes plant cells from animal cells. Prokaryotes, fungi, and some unicellular eukaryotes also have cell walls. The cell wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. Plant cell walls are made of cellulose fibers embedded in other polysaccharides and protein. Plant cell walls may have multiple layers. Primary cell wall are relatively thin and flexible. Middle lamella, thin layer between primary walls and the adjacent cells. And then secondary cell wall, in some cells, added between the plasma membrane and the primary cell wall. Animal cells lack cell walls but are covered by an elaborate extracellular matrix. The ECM is made of glycoproteins, such as collagen, proteoglycans, and fibronectin. The extracellular matrix proteins bind to receptor proteins in the plasma membrane called integrins. The extracellular matrix has an influential role in the lives of cells. It can regulate a cell's behavior by communicating with a cell through integrins. The ECM around a cell can influence the activity of genes in the nucleus. Mechanical signaling may occur through the cytoskeleton. Cytoskeletal changes that trigger chemical signals in the cell. Neighboring cells in tissues, organs, or organ systems often adhere, interact, and communicate through direct physical contact. Plasmodesmata are channels that perforate plant cell walls. Through plasmodesmata, water and small solutes, and sometimes proteins and RNA, can pass from cell to cell. Three types of cell junctions are common in the epithelial tissues. At tight junctions, membranes of neighboring cells are pressed together, preventing leakage of extracellular fluid. Desmosomes, anchoring junctions, which fasten cells together in strong sheets, and gap junctions, which are used for communication. They provide cytoplasmic channels between adjacent cells. A cell is greater than the sum of its parts. Cells rely on the integration of structures and organelles in order to function. For example, a macrophage's ability to destroy bacteria involves the whole cell coordinating, components such as the cytoskeleton, lysosome, and plasma membrane, all in tandem. That's amazing. A macrophage can really extend its phylopodium out to grab bacteria, pull them in, and then engulf them and digest them. Man, such intricate work. I love this. Different parts of a plant cell involve photosynthesis. In the chloroplast, cellular respiration in the mitochondria, and then membrane proteins, which help in the movement across the plasma membrane and cell wall if a cell wall is present. Various other figures of how interactions between the nucleus and the endoplasmic reticulum through nuclear pores. Further embedded proteins inside a cell and those slightly outside. Fascinating. All right, slide 200 of 215. All right, showing how RNA polymerase, the combination of a ribosome, mRNA, and tRNA polymerase, how mRNA is being produced from DNA. That's not a ribosome, that's RNA polymerase is a separate protein. So DNA is being read, mRNA is being produced. Nucleosome, DNA wrapped around 8 histone proteins to help package it. That was transcription. Now translation is where the ribosome, both of its subunits, read mRNA and slowly produce a polypeptide protein chain, one amino acid at a time, as transfer RNA brings an individual amino acid to the ribosome, one at a time. Yeah. One of the motor proteins with their soggy myosin legs. Man, that is just wild. That a protein can move. All right. How different cells can have different functions. Fascinating. Okay, a figure representing how volume and surface area can change. Volume equals 4 over 3 times pi r cubed, r being the radius or half the diameter, and that will change significantly faster than the outer surface area of a cell if a cell were to grow larger. All right, further figures. Almost done, some final figures, and it looks like that's the remainder of new material. All right. Well, once again, thank you for participating in this lecture chapter review. This is chapter 6 of Campbell Biology, 12th edition. Glad to have you all, and thanks for watching. Peace.